In this video, let us learn about the limbic lobe, which is the sixth and the final lobe we will be dealing with in the cerebral hemisphere. The word limbus means border or rim. So the structures forming the limbic lobe form a border or rim around the corpus callosum and the diencephalon, which are seen on the medial surface of the cerebral hemisphere. This limbic lobe is made up of archipallium, paleopallium and neopallium. So at places, the cerebral cortex shows only 3 layers here. It then gradually changes to 4 to 5 layered cortex, finally becoming the 6 layered neocortex or neopallium. The structures forming limbic lobe, along with some of the subcortical structures, together they form what is known as limbic system. So the structures within the cerebral hemisphere, which are forming a rim around the corpus callosum and diencephalon, they form what is known as limbic lobe. The structures of the limbic lobe, when combined with some of the subcortical structures, they form what is known as limbic system. This limbic system is involved in functions like memory, learning, emotions, motivation, survival of the self, and survival of the species. It's an area where olfactory inputs, visceral inputs, and the somatic inputs all converge. So let us find out more details about this lobe. As I said earlier, limbic lobe, the name arises because of the word limbus, means border or rim. During the early embryonic period, the two cerebral hemispheres will begin to develop as two lateral telencephalic vesicle outgrowths. One on either side, they are spherical outgrowths. They will be expanding uniformly around the entire circumference. But soon the central portion, which is going to form the future insular cortex, that will begin to grow much more slowly compared to the areas of the cortex which will be surrounding it. So the surrounding areas will grow faster. They will overhang on the insular part, burying the insula in the depth. And as the expansion continues further, second change will happen. That is, the current posterior pole of this telencephalic vesicle will now make a turn in the form of a C. So it will grow forwards and laterally. So we now have a second portion which was somewhere in the middle, going posteriorly to form the new posterior pole. The original posterior pole will come forwards and laterally to form the future temporal pole. So this is what gives a C-shaped orientation to the cerebral cortex or the cerebral hemisphere. Now this C-shaped orientation is retained for the rest of the life. That is the reason why the cavity of the cerebral hemisphere, known as lateral ventricle, is in the shape of a C. Surrounding that, there are structures which now we are going to talk about as limbic lobe structures. They are taking the shape of a C, as shown by this pink marked area here. There are some output fibers from this limbic lobe itself, known as Fornix, that is taking the shape of a C. There are structures like of the basal nuclei, known as caudate nucleus, that is again taking the shape of a C. So various structures within the cerebral hemisphere will maintain the shape of a letter C. That is because of this initial turning which happened during the early embryonic period. So here we are considering structures forming limbic lobe. You will notice that most of these structures will be taking the orientation of letter C. So first let us identify what structures go on to make the limbic lobe. Anteriorly, just below the corpus callosum, we have what is known as subcallosal area, which continues above and around the corpus callosum as cingulate gyrus. Now the posterior end of the cingulate gyrus continues forward into the temporal lobe, where it forms two parts, a deeper hidden area called as Hippocampal Formation and surface continuation known as Parahippocampal Gyrus. Now Parahippocampal Gyrus you are seeing on the inferior surface of the temporal lobe, that is the tentorial part of the inferior surface. This is the area which is medial to the collateral sulcus. Whereas hippocampal formation will be hidden deeper to the parahippocampal gyrus. So these four structures, subcallosal area, cingulate gyrus, hippocampal formation and parahippocampal gyrus, these are the four structures of the cerebral hemisphere which together are known as limbic lobe. Now coming to limbic system, that includes the structures forming the limbic lobe and some of the subcortical nuclei, which include amygdala, septum, we have hypothalamus, habenular nuclei, anterior thalamic nucleus, and some of the basal nuclei like nucleus accumbens and basal nucleus of Maynard. So limbic lobe structures plus these subcortical nuclei together is known as limbic system. Let us try and identify some of the limbic structures on the medial aspect. Now, one of the special sense pathway is intimately related with the limbic system, that is the olfactory system. So we will begin with identifying olfactory bulb, which is continuing posteriorly as the olfactory tract. Then we are seeing immediately anterior to the lamina terminalis a thin band of grey matter known as paraterminal gyrus. The name paraterminal is because it is immediately in front of lamina terminalis. Now this paraterminal gyrus will continue above around the corpus callosum in the form of the letter C as a thin band of grey matter known as indusium griseum, and covering this indusium griseum there will be two strands of white matter on either side. They are the medial and lateral longitudinal striae. Now the posterior end of this indusium griseum will continue forward into the temporal lobe where it forms the structures of the hippocampal formation, that is hippocampus proper and the dentate gyrus. So the upper indusium griseum connects with the lower hippocampus and dentate gyrus. The junctional area is known as gyrus fasciolaris. Now this is immediately below and behind the splenium. Now the output fiber from this hippocampal formation is known as fornix. See again how this is taking the shape of a C. Only thing is this is going under the corpus callosum, unlike the indusium griseum which was going above the corpus callosum. Now the fornix, when it reaches anterior commissure anteriorly, it divides into two branches, a precommissural column which goes in front of the anterior commissure and postcommissural column which is going behind the anterior commissure. This postcommissural column is what relays into mammillary body, which is a part of the hypothalamus. Now this part will actually form part of the Papez circuit, which we will be dealing later. Then we have some of the fibers of the fornix which will be cutting through the fibers of corpus callosum to relay in the cingulate gyrus. Now that is the structure, or those fibers are called as dorsal fornix. Now coming back to the subcallosal region, immediately anterior to the paraterminal gyrus, we have what is known as prehippocampal rudiment, and further anteriorly we have paraolfactory area. Now this paraolfactory area will continue above the corpus callosum, again in the letter shape of a letter C, with cingulate gyrus, and deep to the cingulate gyrus within its white matter substance we have the long association fiber bundle known as cingulum. Now this cingulate gyrus will continue forwards into the posterior end of this cingulate gyrus will continue with the temporal lobe as parahippocampal gyrus. Now the anterior end of this parahippocampal gyrus shows a part which is separated by the rhinal sulcus, that is named as uncus. We have seen these structures when we were studying the structures in temporal lobe. The cingulate gyrus, junction of the cingulate gyrus with this parahippocampal gyrus is marked by isthmus, which is again the grey matter present behind the splenium of the corpus callosum. Then we have what is known as amygdaloid body. Now this amygdaloid body is one of the subcortical nuclei which is located at the anterior end of the hippocampus or at the anterior end of the inferior horn of lateral ventricle. Its main fiber output is the stria terminalis, which is shown here, again taking the shape of a letter C. Then we have between the corpus callosum and the fornix, we have what is known as septum pellucidum. Now the septum pellucidum and the paraterminal gyrus together constitute what is known as septum or septal nuclei, again a part of limbic system. Now these are the structures which we are going to see, which are all these structures which are labeled here, they all constitute what is known as limbic structures. Now let us begin to study one by one. First let us begin with cingulate gyrus. The word cingulate means belt. It is forming a belt around the corpus callosum. Now the cingulate gyrus shows multiple Brodmann areas. They are areas numbered 25, 32, 24, 23, 31, 30 and 29. This area is quite expansive, so it is divided into 4 subparts. There is an anterior cingulate cortex which is made up of area numbers 25 and 32. Then we have got mid-cingulate cortex that includes part of the posterior part of the area 32 and 24. Plus there is what is known as cingulate motor area, which will be buried within the cingulate sulcus here at its intersection with the supplementary motor area above that. Then the third part, going further posteriorly, is the posterior cingulate cortex. That includes areas numbered 23 and 31. And finally, we have the last posteriormost portion known as retrosplenial cortex that includes areas numbered 29 and 30. The anterior part of the cingulate gyrus receives mainly the connections from the frontal area as well as frontal lobe as well as the various motor areas, whereas the posterior portion receives fiber input coming from the somatosensory, auditory and visual association areas. Now coming to its functions, the anterior cingulate cortex is involved in some of the autonomic functions, including it has an area for bladder control, it's involved in emotions, it's involved in pain and also in vocalization. The mid-cingulate cortex is involved in reward-based decision making, and the cingulate motor area is involved in appropriate motor plan selection. That with the supplementary motor area will select the appropriate motor plan. The posterior cingulate cortex is involved in visuospatial orientation and it is also active during reading. And retrosplenial cortex is involved in retrieval of memory. Cingulate gyrus is actually gaining a lot of importance in the recent years. This is also an area involved in motivation, and this area is implicated in Alzheimer's disease, schizophrenia, anxiety, depression and bipolar disorders. So a lot of studies are happening on this area currently. Now moving on to parahippocampal gyrus. This is seen on the inferior surface of the tentorial part of the inferior surface. So it is seen in the temporal lobe. This is the area which is medial to the collateral sulcus. It has got various subparts. So this is actually the area which forms junction between the allocortex and neocortex. Allocortex is a combined name for both archipallium and paleopallium. So they are continuous with the neocortex at the level of parahippocampal gyrus. From front to back, it is made up of various subparts. Its anteriormost portion is known as entorhinal area or area 28, which is intimately involved with olfactory function. Further behind that, we have uncus, which is separated by the rhinal sulcus. Behind this, we have a small area known as perirhinal area. This perirhinal area has got two components. One portion is adjacent to the rhinal sulcus, so adjacent to the uncus. The second part is lateral to the parahippocampal gyrus, that will be extending to the medial occipitotemporal gyrus, areas 35 and 36. And the posterior most portion is known as posterior parahippocampal gyrus. So what is this parahippocampal gyrus function? This parahippocampal gyrus will receive cognitive and sensory information from all the association areas as well as from the cingulate cortex. Then what does it do? It will send this information for consolidation to the hippocampus. The hippocampus is the one which is going to convert this into memory. And then what does it do? It will retrieve this consolidated information from the hippocampus, which will reach it via the entorhinal area and perirhinal area, and then transfer this information to the association areas and cingulate cortex for storage as memory. So that is the role of parahippocampal gyrus. The third component is the olfactory pathway, but please visit this video on olfactory pathway, or you can go to the site and check out for the olfactory pathway video. Otherwise it becomes a very lengthy description, so we study that as a separate video. Then we have what is known as hippocampal formation. If you take the section of the cerebral cortex as shown by this red dotted line here, we find different components of hippocampal formation. They include hippocampus proper, otherwise known as cornu ammonis, also known as Ammon's horn. The second part is the dentate gyrus. Then we have subicular complex, which include subiculum, presubiculum and parasubiculum. And finally, the entorhinal cortex itself. So let us see how the structures will appear on section. This is the section of the inferior horn of the lateral ventricle, showing the choroid fissure medially containing choroid plexus. Below that, we are seeing the section of the cortex. Within this section, you are seeing how this is an S-shaped structure. It's an unmistakable thing. If you see it once in the specimen of cerebral hemisphere, you will never forget it. So you are seeing the first fissure there immediately beneath the upper part of the cortex, that is the hippocampal fissure. And we are also seeing on the inferior surface the collateral sulcus. So let us first identify the parts forming the hippocampal formation. We are seeing the dentate gyrus here immediately above the hippocampal fissure. It's called as dentate because if you open the lips of the hippocampal fissure and see the surface of the dentate gyrus, it shows broad ridges which resemble teeth. That's why the name dentate gyrus. And immediately above the dentate gyrus, we have hippocampus proper, otherwise known as cornu ammonis. Because it's known as cornu ammonis, there are 4 functional areas separated here. They are labeled as CA1, 2, 3 and 4. CA standing for cornu ammonis. As this cortex takes a S-shaped turn, we get the subicular complex areas, that is subiculum, presubiculum and parasubiculum. It will then continue on the inferior surface with the anterior end of parahippocampal gyrus, where it is going to be named as entorhinal cortex or Brodmann's area number 28. Now, above the cornu ammonis or hippocampus proper, we have collection of fibers, that is, those are actually the output fibers from the hippocampal formation. At this place, we call them as fimbria. They're actually part of the fornix, but at this place, they get the name as fimbria. Then above the inferior horn of the lateral ventricle itself, we have tail of the caudate nucleus and a fiber bundle known as stria terminalis. Stria terminalis is the output fibers from the amygdaloid nucleus. So these are the structures when you take a section in the anterior part of the temporal lobe and you see the formation of hippocampal formation. So what is the function of this hippocampal formation? It does some of the interesting functions. It's involved very actively in learning. So today, if you're learning about limbic system for the first time, the one part of your brain which will be very active at this point of time will be this hippocampal formation. It's involved in memory, especially the explicit memory. Explicit memory is I ask you a question and you answer it back. So you verbally express yourself by recollecting what was in your memory. That is explicit memory, again a function of hippocampus. It's also involved in spatial navigation. In fact, there was that famous experiment conducted on cab drivers in London where it was shown that the hippocampal formation was much larger. So it helps in spatial navigation and probably has a role in emotions. Now coming to the connections of hippocampus. So hippocampus receives olfactory information from the entorhinal area. It receives auditory information from the superior temporal gyrus and middle temporal gyrus. It receives both corporeal and extracorporeal visual information from supramarginal gyrus and angular gyrus, that is area numbers 40 and 39 respectively. and it receives information about object shape and color as well as facial recognition from the inferior temporal gyrus and the fusiform gyrus. So these are basically the various sensory inputs which are reaching the hippocampus, where it is going to be stored as your memory. So what you hear, what you see, or what you smell is getting transferred as your memory. Now coming to how it is getting transferred. Firstly, all these informations will be reaching the entorhinal cortex, which will send direct input to the dentate gyrus. Now this direct pathway connecting the entorhinal cortex to the dentate gyrus is known as perforant path. From here, the fibers are sent from the dentate gyrus to the CA3 area. Those are known as mossy fibers. They are the axons of granule cells present here. From the CA3, the fibers or the collateral fibers are sent back to CA1. They are known as Schaffer's collaterals. And from CA1, the fibers are projected back to subiculum. From the subiculum, the fibers will travel on the surface of the hippocampus proper, forming what is known as alveus. So this pathway is known as alveolar path. Now through this alveolar path, the information is reaching the fimbria, which is actually the output fibers starting from hippocampal formation. From this fimbria, the information reaches or is transmitted to the fornix, reaching the mammillary body. Then from the mammillothalamic tract, it reaches the anterior nucleus of thalamus, finally which will send information to posterior part of the cingulate cortex as well as the retrosplenial part of the cingulate cortex. So this is one way in which memory is sent back from the hippocampus to be stored. The other way in which it is processed is from the subiculum, it will reach back to the entorhinal cortex, and from the entorhinal cortex, it will go to inferior temporal gyrus, temporal pole as well as to prefrontal cortex for storage. So these are the two pathways through which the memory part is being transferred from the hippocampus to various parts of the cerebral cortex for storage. So the main output fiber from the fornix, from the hippocampus is known as fornix. Now this fornix is a fiber bundle which is an exclusive fiber bundle made up of association fibers, commissural fibers as well as projection fibers. So this is the only fiber bundle which has got combination of all the three types of white matter. So let us learn about its various parts. Firstly, the fornix begins from the alveus, which is basically a thin sheet of white matter covering the upper surface of the hippocampus. These are the fibers which will be starting from the subicular area predominantly. So these alveolar fibers will then move to the medial side of the hippocampus, where they collect together to form what is known as fimbria. This fimbria will now ascend below the corpus callosum, that is splenial part of the corpus callosum, where it gets the name as crus. The left and right sided crura will be joined together to form what is known as commissure of the fornix. Now this is the portion which will be behind or below the corpus callosum, below the posterior part of the corpus callosum. Now the two fornices or the crura of the fornix will be traveling forward, where they join with each other to form what is known as body of the fornix. So here the right and left side will come very close to each other to form a single body. Then the fornix continues further forwards till it reaches the level above the anterior commissure, where it splits into two fiber bundles, an anterior portion and a posterior portion. The posterior portion is known as postcommissural column, which will relay in the mammillary body, and the anterior fiber bundle is known as precommissural column, which will relay in the septal nuclei. So these are the various parts of the fornix. In addition, from the upper surface, the fornix will also be sending what is known as dorsal fornix. These will be the fibers which will cut through the corpus callosal fibers to reach the cingulate gyrus. Now that is why this fornix is known to be having all the three types of fibers. That is, it starts from the hippocampus of one side, and through the dorsal fornix, it connects with the cingulate gyrus of the same side. So connecting two areas of the same cerebral hemisphere, it is association fibers. The right and left fornix are joined together through the commissure of the fornix. That is, the fibers which are starting from the left-sided hippocampus will ascend through the fornix of the left side. They shift to the right side at the commissure of the fornix. They go in the opposite direction to reach the right-sided hippocampus. So these are the commissural fibers. Then the rest of the fornix will travel forwards, where it is relaying in the mammillary body, which is part of the hypothalamus. So from the cortex, cerebral cortex, it is projecting to one of the subcortical area. Therefore, it is projection fibers. So within the fornix, we have association fibers, commissural fibers, as well as projection fibers. Long ago in the previous century, Pape proposed a circuit which he thought was involved in controlling the emotions, but soon it was found that actually the circuit is involved in laying down the memory. So the Pape circuit begins from hippocampal formation, where it is sending its output through the fornix to the mammillary body. From the mammillary body, the projection is to the anterior nucleus of thalamus through the mammillothalamic tract, otherwise known as Victi azure. And from the anterior thalamic nucleus, it sends output to the cingulate gyrus, where the fibers will be passing through the internal capsule. And from the cingulate gyrus, through the cingulum, which is the association fiber, the information reaches back to the hippocampal formation. So this is the basic Pape circuit. Initially, it was thought to be involved with emotions, but currently it is understood to be involved with memory, especially the associative part of the memory. Now, more recently, some more structures are added to this Pape circuit. So from the association cortex, there will be direct input to the cingulate gyrus, both input and output to the hippocampal formation. So that is how the association cortices having a role with memory. Then hippocampal formation will project to the hypothalamus either directly or via the amygdaloid nuclear complex, and the hypothalamus in turn will project back to the prefrontal cortex. This is another few parts which are involved in memory. So all these portions which are shown in green color, they are actually involved, or they are included in the Pape circuit more recently, but the original Pape circuit is what is shown by those 4 structures in the center. Now coming to other structures like septum. Septum, when we say a part of the limbic system, we mean 2 parts. The septum pellucidum, which is a sheet of white matter containing collections of neurons or nuclei. That is, it extends between corpus callosum and the fornix. So that part is known as supracommissural septum because it is above the anterior commissure. Then we have paraterminal gyrus, which is immediately anterior to the lamina terminalis. Now that also contains collection of grey matter which will form part of the septal nuclei. Because it is immediately in front of the anterior commissure, it is known as precommissural septum. So these 2 parts together, that is septum pellucidum and paraterminal gyrus together, we call it as septum. So coming to the inputs for this septum, it receives inputs from olfactory tract through the medial olfactory stria. It receives input from amygdaloid nucleus through diagonal band of Broca. It receives input from hippocampus through the fornix, and it receives information from the monoaminergic neurons in the brainstem through the medial forebrain bundle. 2 main outputs. One is the stria medullaris thalami, which sends its information to the habenular nuclei. This pathway is involved in sleep-wake cycle. The second one is the septo-hippocampal pathway. So these are the fibers starting from the septal nuclei. They will travel within the fornix in the reverse direction to reach the hippocampus, and these fibers are involved in episodic memory. Stimulating septal nuclei results in sexual sensation in human beings, and lesion to the septal nuclei in case of experimental animals resulted in septal rage. Now coming to the next part of the limbic system, that is amygdala. Now this is located in the anterior end of the inferior horn of lateral ventricle, where it forms the anterior surface, medial surface and superior surface of the tip of the inferior horn. It has got 3 subgroups of nuclei: lateral group, central group and basal group of nuclei. Lateral and basal together form what is known as basolateral part of the amygdaloid nuclear complex. The central group of subnuclei will form what is known as cortico-medial group of nuclei. So what are its inputs? It receives auditory information from the superior temporal gyrus, tactile information from the parietal cortex, visual information from the occipital cortex, olfactory information from the piriform lobe, mnemonic or the memory related information from the hippocampus, nociceptive or pain related information from the midbrain reticular formation, cognitive information from the orbitofrontal cortex, and attention related information from the basal nucleus of Maynard. Its main output is through the stria terminalis, or the second pathway is the ventral amygdalofugal pathway. So what are its outputs? Either it sends an output to the periaqueductal gray in the midbrain, that is for anti-nociception, or to the nucleus ceruleus, which is involved in arousal, or to the medullary reticular formation, especially the noradrenergic neurons, that is for resulting in tachycardia and hypertension. It sends output to hypothalamus and dorsal nucleus of vagus, which is involved in bradycardia as well as fainting response. Sends output to the parabrachial nucleus, which is going to result in hyperventilation. Or it sends amygdalocortical output to almost all association areas in the cortex as well as to prefrontal cortex. It also sends information to nucleus accumbens, which is part of the basal nuclei, responding to basically all pleasurable sensations. So what is the function of amygdala? It is involved in emotion of fear. And bilateral lesion to the amygdala will result in what is known as Kluwer-Bucy syndrome. Now, Kluwer-Bucy syndrome, here the patient has an excessive sexual and hunger drive. He does not recognize objects visually, so he wants to keep feeling them and even examine them orally. So these are the features of Kluwer-Bucy syndrome, which is because of the lesion to bilateral amygdala. Now, on the clinical side, they use a term known as medial temporal lobe, lesion to the medial temporal lobe, seizure arising from the medial temporal lobe. So what do they mean by medial temporal lobe? Three structures together form medial temporal lobe: Parahippocampal Gyrus, Hippocampal Formation, and amygdala. So together it is going to form what is known as medial temporal lobe. Now coming to functions of limbic lobe. One, it is involved in memory. So let us find out about the various types of memory. There is what is known as short-term memory. This is holding any new information very briefly in mind. Like when you are dialing a number, you just remember the telephone number at that point of time. So that is the short-term memory. Contrary to this, we have long-term or remote memory. So here information is stored so that you are capable of retrieving it at appropriate moments. Two types are there: explicit or declarative memory. So this is verbally expressed. If in case you're expressing it in an autobiographic or in like narrating an experience, it's also called as episodic memory. Or it could be implicit or procedural memory, where it is the memory of performing a motor skill. Then we use a term as working memory. This is simultaneous retrieval of several items from long-term memory for a particular given task. A classic example is when you are driving through a familiar lane. So you are performing a motor skill of driving. You're trying to recollect what are the structures on the roadside because you're familiar with that road. Trying to remember where you have intersecting roads or even a traffic light, so you want to slow down. You may be even remembering or recollecting whatever person you may have met or where to stop your car to reach a particular place. So a lot of things will be going on when you are driving through a familiar lane. You are recollecting only that much part which will be relevant for that particular patch of driving. So that is the working memory. Then there is a term known as consolidation. This is the process of storing new information in long-term memory. So what happens in the memory of limbic lobe if there is any lesion here? Anterograde amnesia results whenever there is bilateral damage to anterior part of the hippocampus, that is absence of conscious recall of newly acquired information. That is specifically loss of explicit or declarative memory. It does not affect the procedural memory. So what do we mean by this? You keep 10 dishes on a dining table, the patient will be able to recall every dish because he has seen it in the past. It's not a short-term memory for him, so he can identify individually what each dish is. But ask him what did he have for his breakfast that morning, he won't remember. So short-term memory is lost. That is what we call as anterograde amnesia, because this is a newly acquired information which he cannot store. Then we have lesion to Papay circuit or diencephalon. This will result in loss of relational memory. That is, when I say diencephalon, it is predominantly to the anterior nucleus of thalamus. So here, patient cannot connect one thing to the next in a form of relation to establish a storyline. So he tries to fill up gaps in his memory by putting a new story there, what is known as confabulation. That's called as Koroskov's lesion or Koroskov's syndrome. Then we have certain areas which are involved in particular function, that is left-sided hippocampus and the dorsolateral prefrontal cortex encode new material in language function, whereas right-sided hippocampus and right-sided inferior parietal lobule are engaged in spatial tasks. So if the London cab driver was being assessed, he will be having his right-sided hippocampus more active because that will be relating to the road maps. Then coming to left anterior hippocampus, especially the anterior part here, that will be active whenever you're reading something new. Like right now you're reading what is written in the screen here. So that's a new topic, let us say. So you will be having the left-sided anterior hippocampus active. Let us say you revisit this video another 10 times. So when you're reading the 10th time on the same page, it will be pushed to the posterior part of the hippocampus. With familiarity, the posterior part of the hippocampus becomes active. Lesion to the medial temporal lobe will result in deletion of all personal memories. That is, it's an ego-centered. So whatever happened to you, that will be forgotten. Whereas lesion to the visual association cortex will delete all recall of all the impersonal past scenes. So some accident happened to some other person, that will be stored in the visual association cortex, but something happened to you will be stored in the medial temporal lobe. So again, loss of memory of which type will tell you where could be the likely site of lesion. Then the dorsolateral prefrontal cortex, that is the area which is concerned with working memory because it gets input from various association cortex. So it will be using this information to recall from the related association inputs. Now coming to another function of limbic lobe, that is emotions. By definition, emotions is any feeling state. That is, you may be feeling fear, anger, excitement, love, hate, happiness, whatever. So that is a state of feeling is emotions. The way in which or the sequence in which it happens is there will be a stimulus. It could be an actual stimulus or it could be an imagined stimulus which will result in a feeling. So either you see somebody and therefore you feel something, or you imagine that somebody and you feel something. So real or imagined stimulus which will result in a feeling. Now the feeling is then expressed or there is a response to that, which could be visceral response, like you have increased heart rate or flushing of the face, dilated pupils, any of these visceral responses. There could be somatic responses. You may flare your nose or try to get into fist fight or any of the somatic expression, that is second type. The third could be verbal expression. You want to express your feelings by vocalizing them. Or there could be a cognitive response wherein you're shifting the information to the orbitofrontal cortex will say, okay, no need to express or only express it minimally. So there is a cortical control, that is a cognitive type of response. So any emotion is first, it begins from a stimulus. You go through the state of feeling it and then you decide on what type of response will be there. Then we use the term affect. Affect is the observable display of feelings. So what you're feeling inside probably is not understood by the others, but when it is displayed by any of these responses, visceral, somatic, verbal or cognitive, that is what is known as affect. So what are the structures involved in emotions? All the connections between different parts of the limbic lobe, connections between limbic lobe and the orbitofrontal cortex, with the hypothalamus, amygdala, as well as midbrain. So those are the structures which are involved in expressing emotions, feeling as well as expressing emotions. The main fiber bundle involved in this function is the medial forebrain bundle. Now this is again a projection fiber bundle which has both ascending and descending fibers. This will be passing through the lateral part of the hypothalamus. It connects orbitofrontal cortex, septal nuclei, amygdala and hippocampus anteriorly with midbrain and pons posteriorly. So this is a structure fiber bundle which starts from the cortex, pass through the hypothalamus, which is a part of diencephalon, and reach the brainstem. So that's known as medial forebrain bundle, which is very important fiber bundle in case for the function of emotions. So thank you very much. Hope you enjoyed this video. Limbic lobe does fantastic, very interesting functions. In fact, there are a lot more details. I've tried to cut it down, but still this video has been a bit long. Thank you for your patience. You can visit this site for other neuroanatomy videos.